right, well, hello, and thank you all for coming along today. Before we get into the talk, exciting talk by Sethi, I just wanted to take a quick 5 minutes to introduce you all to both our 3' and our brand new 5' single-cell CRISPR screening assays. We'll begin with the question that many people ask, and that's if I'm already doing either bulk pooled or bulk array CRISPR screens, why would I want to perform a single-cell CRISPR screen? The most obvious reason is the ability to directly link tens to hundreds of CRISPR perturbations to their corresponding impact on the whole transcriptome with single-cell resolution. But beyond this, you also gain scale and speed compared to the more traditional screening approaches, as clearly outlined in the graphics on this slide. Before I dive straight into the technical details of our 3' and 5' CRISPR assays, let's take a few steps back and look at the Chromium single-cell platform as a whole. As you can see, the Chromium platform consists of an instrument called the Chromium controller, which is shown here on the slide. It's about the size of a shoebox and fits on your lab bench. Into this instrument, you feed these disposable microfluidic chips, also shown on the slide, and then onto those chips, you can run one of 4 main assay types, all of which are listed here. And regardless of the target analyte, the key output from all these assays is the ability to characterize cellular heterogeneity. I do also want to highlight that for all these assays, we're ready to use robust workflows, including demonstrated protocols for various sample types and turnkey software tools. So if you're new to the world of single cell and want to apply it to your research, but feel intimidated about where to begin, then we have solutions for you across the entire spectrum of the workflow for every one of our assays. But let's move on by taking a closer look at our 3' CRISPR assay, which is an add-on capability to our single cell gene expression product. The target analyte from the single cell gene expression kit is mRNA, where we're capturing transcriptome-wide 3' digital gene expression from single cells. So that's definitely the core of the assay. We can actually extend the menu of analytes being detected from a single cell using what we call our feature barcoding technology. You can also detect cell surface proteins in addition to transcriptome-wide gene expression from the same single cell. This is achieved by adding a DNA barcode, or as we call it, a feature barcode, to an antibody that will then bind the cell surface protein. Using a similar approach described on the previous slide, you can also detect CRISPR perturbations alongside transcriptome-wide expression from the exact same single cell. This works by incorporating a small piece of DNA into the guide RNA so that it can be captured by the assay biochemistry, and we'll explore that further on the next slide. So from the 3' CRISPR assay, you get a readout of what the guide RNA was, which tells you about the CRISPR perturbation that occurred in the cell, but you're also getting a transcriptome-wide snapshot of the functional impact of that perturbation. As I said on the previous slide, you need to incorporate a small section of oligo into your guide RNA in order for it to be compatible with the assay. There are 2 possible locations for integrating the capture sequence, and there are 2 different types of capture sequence to select from. So this means there's a total of 4 different possible configurations that you can have for your guide RNA. We have guidance on our website as to how to select which guide configuration may be best for your studies. But at this point, I do want to reiterate the specific point about remembering that for the 3' CRISPR assay, you need to modify your existing guide RNAs to include this capture sequence. And this is a very important point when contrasting this assay to the 5' CRISPR solution. So as I said at the start of the presentation, we recently announced that alongside our 3' CRISPR assay, we'll be launching our 5' CRISPR assay. And this will be an add-on capability to our single-cell immune profiling product. But many people have asked, since we've had that announcement, why would we have a 3' and a 5' version of the CRISPR assay? What's the main difference? As we just saw, the key limitation for 3' CRISPR is that you must insert a known oligo sequence into the guide RNA in order for it to be detected within the assay biochemistry. However, for the 5' CRISPR assay, you don't need to modify the guide RNA in order to detect it. So you can take existing Cas9 guide RNA libraries, which are the most commonly used type in bulk studies, and rapidly deploy these in 5' CRISPR with no redesigns or reoptimization. Also, just like its 3' counterpart, the 5' assay maintains the ability to measure gene expression and guide RNA for the same single cell, but now you're also getting the added benefits from the single cell immune profiling assay, being able to also layer on full-length paired VDJ profiles from T cells and B cell surface receptors. The 5' CRISPR assay is expected early 2022, which is just right around the corner. For those of you who want to run single-cell CRISPR screens but didn't want to mess around with your existing guide RNA structures, then this product will be ideally suited. So to summarize, the key take-home from this short presentation is that Using either 3' or 5' CRISPR assays, you get a readout of what the guide RNA was, which tells you about the CRISPR perturbation that occurred in the cell, which also getting a transcriptome-wide snapshot of the functional impact of that perturbation. This means with single cell resolution, you can profile hundreds of different gene perturbations by detecting single guide RNAs and directly linking each perturbation to an expression phenotype. Also, unlike the 3' CRISPR assay, with our new 5' CRISPR assay, you can take your existing Cas9 guide RNA libraries and use them with no redesigns or reoptimizations. So with that, I'll hand it over to Sefi, and he can take you on a deep dive into some single cell CRISPR data.
Thank you, Jeff. Let me just share my screen. Okay, thanks. And thanks, Jeff, for that introduction. It's just a nice way to introduce what I'm going to talk to you today, which is about the application of pooled CRISPR screens with single cell RNA sequencing as a readout. So kind of, and maybe I should just mention before that everything I'm presenting to you is a very close collaboration between my lab, the Monash Functional Genomics platform, and Phenomics Australia. So Phenomics Australia funds a lot of these operations, doing these kind of experiments, and I'll talk to this also in the end. We do have opportunities, thanks to Phenomics Australia, to help anyone who wants to do these experiments for very reasonable amounts of money. So the motivation behind doing these kind of CROP-Seq experiments is, like Jeff just said now, is the fact that you could do them quickly and for relatively low cost. So the idea is that we all like to use pooled CRISPR screens because they are easy to use, they're very quick, we could generate CRISPR libraries very, very fast and robustly, and then measure phenotypes. Most of the phenotypes that we typically measure in a pooled screen are these very simple phenotypes, kind of looking at things like proliferation or maybe reporter activity. But here, what we want to measure is something more complex, and basically using a pooled screen to measure mRNA profiles. So the idea is that we want to take a pool of guide RNAs, infect a particular cell, and then use the Chromium 10x to separate the cells, and then based on a readout, get both a readout of the mRNA profile of the cell as well as the guide RNA identity. So we could say guide RNA number 1 knocked down gene X, and that caused these changes in mRNA expression. Of course, you could do this just one at a time in an arrayed format. The problem is, again, this will take longer, it would be more expensive, with the advantage of course, having a better resolution than you would get in 10x. And I'll show you some of this, some of also the issues that this technology has. One thing that we get quite asked a lot is about this technology. And basically, there are two main publications that describe this technology, one called Pertrub-Seq and one called CROPSeq. I just want to talk about a little bit about the differences between them, because there are kind of important differences between them. So the first publication from Aviv Regev's lab described a technology called Pertrub-Seq. In Pertrub-Seq, and both of these technologies, by the way, there's no need to add additional sequences to your guide RNA, but the way the vector is built is kind of in a different way. So in the Pertrub-Seq, what you do is you basically put a U6 promoter, which is a pol3 promoter, transcribed in your guide RNA, which will knock out or suppress your gene of interest. And then downstream of it, you need to have an E1F alpha promoter. So this is a pol2 promoter transcribing a gene and a guide barcode. So this is just a random barcode, which has a poly A sequence, is captured by the 10x machine and could be read. So when you have this vector intact, basically you would infect your cell, the guide RNA would knock out your gene of interest, and because of this poly A sequence, you will identify the identity of the guide. The main problem that this methodology has is what we call template switching. So what happens is, because of this very long distance between the guide barcode and the guide RNA, you get this random, when you make lentiviruses out of these libraries, you get this random switching between guide RNA and guide barcodes. And it's possible, but it's very messy to use. Furthermore, it's kind of hard to make these libraries. So these libraries are not easy libraries to make. Just a bit after this publication was published, this publication from Chris Bach's lab published this methodology called CROPSeq, which is a really cool trick that basically avoids all these problems. And what Chris Bach's vector does is, instead of having a separate U6 and an E1F alpha promoter, what it does is you basically put the E1F alpha promoter behind the U6 promoter. And what that does is you basically get two transcripts in this cell. You get transcript number 1 is this one right here, the U6 promoter, which transcribes a guide RNA. The second vector, the second transcript you get is this long transcript, which contains the puromycin, it contains the U6 promoter, as well as the guide RNA sequence, followed by a poly A tail. Now, this sequence is actually captured, it doesn't do anything in the cell, but is actually captured by the 10x machinery and could be used to identify the guide RNA identity. So the cool thing about this is that all you need is to clone your guides into this vector, which is very similar to all our other CRISPR vectors, just the fact that the E1F alpha is behind the U6. And when you do this, you could, this works very well. It's very simple to use and could be very robustly used. And I'll show you now a few examples of how we do this. So first, kind of the experimental outline of such an experiment. So the first thing we do is, of course, we generate the libraries. Typically, the libraries are between 200 to maybe 1,000 guide RNAs, depending on what we'll do, and I'll show some examples. Then we infect our cell of interest, and we can do this at low MOI. I'm going to show you sometimes we're going to do it at high MOI, so that the high MOI will determine how many guides per cell you have. Puromycin select. Typically, we let the cells stay for about 10 days in culture, and then we do the 10x single cell separation. We found that overloading gives us better results because we're okay with having some doublets. We could pretty easily get rid of them. A very important point to do is we do PCR enrichment for the guide barcodes. So if you don't do PCR enrichment, then what you get many times is that you have a lot of cells without any guides inside of them. And then we sequence, and we found that the MGI sequencing is also cost efficient and gives us very robust and good results. And we do this about 400 million reads per 10,000 cells. And data analysis, which I will show you in a minute. So the first experiment that I'm going to show you is how we use CROPSeq to look at mechanisms that are associated, mechanisms and drugs that are associated with particular genes. So this, in this example, I'm going to show you genes that are related to breast cancer, but it's this purpose of this talk, it's not really important. The more important is to look at the at kind of how we are doing this. So this library that we have here contains 185 guide RNAs. It targets 55 genes, so we have 3 guides per gene, plus 20 negative controls. So the negative controls are guide RNAs that target just GFPRFP, so non-human genes. This library is infected in B-ADT5 cells, so that's a memory cell that expresses wild-type Cas9. We do this at low MOI, so we want to have 1 guide per cell, 10 days in culture, and then we go through our 10x working our work scheme. So first of all, this is how the results look like. This is what we get when we run the cell ranger. So what you can see is the number of cells. We get here about 15,000 cells, which is a good run for us. We want to get, these are what we usually kind of aim for. We get about 2,500, sometimes you get 3,000 about genes per cell, which is very typical. So this is a run that worked very well. This looks very good. But what I want to really make you pay your attention to is when we look at this CRISPR application, which we could use with the cell ranger. And what you could find here is when we look at the number of guides per cell, this is what we typically will see. So you'll see cells with 1 guide. So remember that we were aiming to have 1 guide per cell. So we have all cells have at least 1 guide, but almost all the cells, 99.9% of the cells have more than 1 guide. Now we have a lot of hypotheses why this happens. And one of them is because of dead cell. We don't really know for sure, maybe because of dead cells in the tubing that cause some guide RNAs to be dispersed somehow. We're not really sure how it happens. But what we do know is that in order to actually identify cells with a particular guide, what we need to do is look at the most robust guide RNA. So this is what it looks like when you're looking at the distribution of a guide per cell. So here you're looking at a plot showing you the percent of reads in a particular cell with guide RNA number 1 and guide RNA number 2. And what you could appreciate that the majority of the cells have one dominant guide. So guide number 1, the most dominant guide, that's most of the cells. There are a few that have both guides in a similar manner, but most have one guide. And for this, we developed this kind of plot here, which is really important in these experiments we found, to look at the threshold of UMIs that you want to use to determine what we call a guide in a cell. So what you see is if you use a very low threshold, then you'll have kind of a lot of guides in a cell. So most cells will have more than one guide. Typically, we use a threshold of 10, meaning only above 10 UMIs for a guide do we call this a guide RNA in the cell. And when we use that, you see here that most of the cells that we have only one guide in them. And that's really important, and you'll see that we use this quite a bit in this analysis, because without this, it's really hard to determine what guide is in your particular cell. So the next thing you want to do as a quality control, you want to see how well is your gene of interest knocked down. So here in this plot here, you're looking at the gene targets, and the dot to represent the knockdown. So each dot represents a different guide. And what you could appreciate that some genes are fairly well knocked out, and others, if you look at the gene knockout of the target gene, it's basically nothing. It's basically still here. And the reason for this is not because they are knocked down. This is another limitation of the 10x. It's more because of the detection. So some of these genes have very low detection value. So for example, here, you could see that these 3 genes here, they're just they're not very abundant genes, and they're detected at very low levels in the 10x. And then if you compare kind of for this lowly detected genes, if you compare control to the gene targeting, you don't see any knockdown, as opposed to the genes with high detection rate that we see very nice knockdown of the gene. But despite the fact that you don't see knockdown of the genes, we actually, if we look at the function of the gene, we do see that they are giving us correct phenotype. So for example, here, Raptor, right, is a very well-known component of the mTOR signaling pathway. And if we look at the genes that are going up and down when you knock out Raptor, they make sense. So we get kind of mTOR signaling pathway. That's what you would expect to see. Or similarly, if you look at APC or casein kinase 1 alpha, so these are 2 negative regulators of the Wnt signaling pathway. If you look at the knockdown of them, you basically see nothing. It doesn't look like they're knocked down. But when we look at known markers of the Wnt signaling, axin 2, so these are out of all of our genes, these are the only one who upregulated axin 2, which is what you would expect from a gene that activates the Wnt signaling pathway. So to conclude this, what we found is that using the actual target knockdown as a matrix is not a very good one, because that's one of the limitations of the 10x, that it doesn't identify all the genes. So we're more looking at phenotypes that come out of the genes and not really at the actually target gene knockout. But this is what we could do with this. So if we take these genes that we profiled, and the first thing we do is hierarchical clustering, right? And this is just based on gene expression. So we take the gene expression, that is genes that go up and down for each one of the genes, and based on that, we cluster all of these genes. What we find is that we get a lot of well-known signaling pathways that show up. For example, all the components of the PI3 kinase signaling pathway clustered together in this very tight cluster. So PI3 kinase, mTOR, Raptor, right? Wnt signaling, again, clusters together, TGF-beta, cell cycle. So this is a very well and nice way to look at pathways and genes that cluster together. So we could find, of course, known gene, and of course we find some new things that we didn't know before, and this gives us ways of hypothesis of how to look at these genes and potentially what these genes are actually doing. Another thing that we could do with this kind of data is look at drug repurposing databases. And here we use a database called iCLUE, or the Drug Repurposing Hub. This Drug Repurposing Hub is published by the Broad Institute, and this is a hub that has in it more than 1 million gene expression signatures. So they were created using a methodology called L1000. And what you basically have is a gene expression changes mediated by a lot of different small molecules. So what you could actually do, and it's very simple to use it. This is what the platform looks like. You could take your target genes. So we could take the target genes of interest that we want to do. We could put in the genes that are going down, the genes that are going up by that knockdown, and then query that against the entire collection of iCLUE. So we could find small molecules that are similar or dissimilar to those gene signatures. So for example, if we look at positive control, so known genes, so here we're looking at, for example, components of the PI3 kinase pathway, and what you see, these are positive components, and you see that the signatures that are mediated by these gene knockouts are very similar to known PI3 kinase inhibitors, right? Same thing for EGFR, NF2, or P53, right? So we're getting good signatures. So even though the expression profile is not full, it's good enough to connect them to known compounds. And of course, when we look at our new genes, we could find new types of compounds and new classes of compounds that connect to those genes, and that gives us hypotheses of compounds that we could potentially use to target these genes, to explore, or to study these genes of interest. So that's one CRISPR kind of approach that we have. So this is infection at low MY. This basically replaces the use of kind of standard RNA-Seq, which you could do at much higher throughput for low cost and less time. The second approach that I want to talk to you is an approach called CRISPR-QTL. So this approach is, again, same kind of idea of this CROP-Seq, but here what we want to explore is, what we want to look at is enhancers. So the question we're asking here, we have a lot of candidate enhancers. So these are genomic elements that are regulating genes. And we want to ask, what are the genes that are regulated by a particular enhancer? And the way we do this, we use a form of CRISPR called CRISPR-I. So here we use not wild type Cas9, but this is dCas9. So it's a mutant Cas9. It could bind to DNA, but no longer cut DNA, fused to CRAB, a transcription repressor. With the idea is that if this complex is bound to an enhancer, the enhancer will loop to its gene target and inhibit its expression. Now, on top of this, the way we do this experiment as different than what I showed you before, what we do for our normal CROP-Seq. Here with CRISPR-QTL, we infect the cells at high MOI. So this is a cool way to even reduce cost by more. So we have multiple guides per cell, and the idea is that if we average across a particular perturbation, we will get the correct expression profile, because we're only interested in the genes that are next to the candidate enhancer, but not the complete genome. In the original paper, they described doing this with an MOI of 20, so 20 guides per cell. And we thought that's a lot. But so we did an experiment here using a library containing 1,000 guides. And we're using this at an MOI of 5. So we have about 5 guide RNA per cell. It's not 20 guide RNAs per cell. Again, 10 days in culture, and then we measure gene expression. So again, we get very nice number of cells, guides per cell. And here is the plot that I showed you before, and I think you could appreciate here the difference that we have here many more cells. And again, we usually use a threshold of 10 UMIs in order to determine which what guide per cell. And of course, we have here a lot more guides per cell, as you would expect. So most of the cells have more than 2 guides in them. A lot of them have even 5 and more. Okay, so the first thing we do is quality control. We want to look at known promoters. So here we have 100 guide RNAs targeting 50 genes. So these are not enhancer targeting, these are gene targeting. They target the transcription start site of known genes. And when you look at all these guide RNAs, you can see a very nice inhibition of the target genes. This methodology works, even though we're doing this high MOI. So it's almost surprising to me that it worked so nice, but it works. The second thing is to now we want to determine the enhancers that are regulated by a target gene. And there's been a couple of different algorithms, analytical platforms, and the one that we found worked best for us is one called CROPSeq. So here's when you're doing a QQ plot looking at known CROPSeq experiments. And what you see that the QQ plot for negative controls differs from the expected. And this CROPSeq algorithm corrects for that by using a binomial z-value distribution and then resampling and correcting for this null distribution. And when you do that, that kind of fixes the problem. We found that this works very well for our experiments. So here's an example of an enhancer and the genes that it targets. So in this case, you have here, right here in red here, this is the enhancer. And we have 10 guide RNAs that are targeted to this enhancer. And each line here represents a z-score, we plot lines with the z-score -1 to a gene in the vicinity of that enhancer. And what you could see here, we usually typically call a gene with more than 3 guides that have a z-score less than -1 as significant. So what you could see is that this enhancer here is binding or targeting 3 different genes, ATF7IP, EMP1, and GPCRA5. All the rest have, maybe 1 guide that shows some inhibition. And you'll always have some noise in these kind of experiments. ATF7IP was the strongest one that we found, and this is what it looks like. So you can see here, this is the enhancer. This is a closer look at that. So basically all of the guides in this enhancer are binding and inhibiting ATF7IP expression. And this is also supported by using high-cheap or TAGx or other ways to determine this interaction. We further wanted to look at this. So to validate this, we first used CRISPR-I. So this is not in a pooled format, but just in a normal format. And you can see that when we target the enhancer, ATF7IP expression goes down. Or we could use a luciferase assay in which we take this enhancer and we insert in either variants in that enhancer that inhibit its activity. And we see that this works. Again, this enhancer works really nicely. This is just to show you that we could do this for many other enhancers, and this CRISPR-QTL kind of finds candidate out of the enhancer. So here we have an enhancer, and we find different genes in these enhancers that this enhancer, at least based on CRISPR-QTL, binds to. And what was nice is when we've done a functional screen for all of these enhancers, the functional screen agrees with the CRISPR-QTL, and for each one of them, we find at least one of the genes in these enhancer which are functional. That was nice to see that out of 6 enhancers we looked for, all 6 of them we found there was an agreement between the functional screen and the CRISPR-QTL. So to summarize, I showed you that CROP-Seq is a powerful way to measure mRNA expression. You could do this at very high throughput for a very reasonable cost. I showed you that you could use it to identify mechanisms of actions and gene targets. And I finally showed you that CRISPR-QTL, or CROP-QTL, but we'll call it CRISPR-QTL, could be used for identifying genes that are regulated by enhancer. And even that we do this at very high MOI, we could still very robustly identify genes that are connected to a particular enhancer. And with that, I will end. I thank people in my lab, as well as the bioinformatics that was done at the Monash Bioinformatics platform. And I would just say that Phenomics Australia has funded us in the Monash Functional Genomics platform to allow people to do this for a very reasonable cost. So if you're interested, please contact me, and we're happy to talk of how we could help you do these kinds of experiments. And with that, I will end. Do you have questions?
Hi, Seppi, sorry. Thanks. That was quite very broad and a very good overview. I have one question about the infection of the cells when you are using MOI of 5. How do you, because if you have an infection that is higher and they infect several cells, how do you, when you have your data, how do you control of several guides into the same cell? So
we don't. The basic idea behind that is that you have the same guide in multiple cells, and that basically averages out. So although in one cell you have, so in every cell you'll have a different combination.
Yeah.
And then basically it averages out. So I wouldn't use this methodology if you'd want to basically look at global gene expression. It's probably, you know, that wouldn't be very useful. But especially for when you're looking at enhancers, you're only interested in the genes in the vicinity, and that seems to work pretty well.
So yeah, because you then in that way can control the expression just locally.
Yeah.
And then you don't have,
we
like,
initially thought it would have like tons of noise because of this, but it doesn't seem to be extremely noisy. It seems to work fairly well.
Okay. Yeah, thanks.
So there was a question in the chat. If more than one enhancer are targeted in a cell, how can we distinguish the downstream effect of these enhancers?
Well, the downstream effect, you mean, if you mean like functional, would be very hard. So this experiment won't tell you anything functional about a cell. All this experiment will tell you is this enhancer regulates this gene. But like if you're looking at downstream effects, that this would not answer that. So it's but to say this is kind of a major question, especially like in GWAS studies, for example, which a lot of them are enhancers. It's very difficult to determine what enhancer regulates what gene. And this seems to be pretty robust. And I would say it looks like it's, in a lot of ways, it's better than a lot of the methodologies that are currently used, like Hi-C, for example, or ChIP-seq, because the resolution here is much better. So when you use, for example, Hi-C, you get kind of a lot of genes that interact with every enhancer, and some of them are real, some of them are not. Here, you typically get a much lower number of genes, and they typically validate. So I think that the resolution of this is much better than what you get with current methodologies.